Hello students, welcome back to the biology class. So in the last class I gave you, or we started the concept of the Morgan's experiment. We learned why he chose the fruit fly. Fruit fly is also called as Drosophila melanogaster, and because of his excellent experiments with this material, he is also called as the father of experimental genetics. So Thomas Hunt Morgan, or T.H. Morgan, is also called as the father of experimental genetics. And remember, his students also were excellent in the experiments which they performed after Morgan. He is also called as the flyman in the field of genetics. He is also called as the flyman because he used fruit fly. Now, why he chose fruit fly in the last class we have learned. We understood that the fruit fly size is different. Female is bigger in size, male is smaller. Because they are unisexual, experiments are easier. With the help of the hand lens or the dissecting microscope, easily the distinguishing, contrasting traits of characters can easily be distinguished. Then their lifespan is very short, so quickly hundreds of progeny individuals are formed just with the two individuals. So two parents you need and hundreds are there with you within the time span of 14 to 15 days. So a lifespan is very short, so experiments goes faster. And then you don't need lots of space or farm or something like in the case of Pisum sativum. Here you need a small box, and with the help of rubbish material like molasses, or you have fruit peels, or you have potato peels, we can easily rear the fruit fly. So that's why Morgan chose fruit fly. Then I explained to you that how his results came different, and that's why he came up with the concept of linkage. And I explained to you linkage is the physical association of two genes, or when two genes are together on the same chromosome, they are called as linked gene, L-I-N-K-E-D, linked genes. Now there were some points regarding the ratios I was explaining to you, so let me revise that quickly to you. See, when we talked about the inheritance of two genes, that Mendel's experiment, we understood these ratios and all. So I was explaining yesterday with my notes that the same thing was explained by Sutton and Boveri, because Sutton and Boveri came up with the idea of chromosomal theory of inheritance. So they stated that it is not the genes which are moving, or the factors of Mendel, because Mendel called them as factors. So they stated that it is not the factors which are moving, but they are the chromosomes which move, because factors are positioned on the chromosomes. Now we all know today, because by the time things became very clear with the scientists, Mendel could not prove these things because experimental things, the techniques and all, biolabs and all were not so high-tech and advanced. But later it became all clear that chromosomes move during gamete formation, and because chromosome move away, the homologous pairs will move away from each other. So that's why reductional division takes place, and that's why only one out of the two will go into one gamete. So why gametes are called pure? Because every gamete will get only one out of the homologous pair, and that also one sister chromatid will go. That means one daughter chromosomes. So in the same manner, Mendel also talked about that during gamete formation alleles separate. So that's why they thought that that's why they came up with this concept of chromosomal theory of inheritance. And according to them, when chromosomes move during gamete formation, alleles on them will also be moving. And when random fertilization occurs, they again come close and a pair is formed, and zygote again becomes diploid in nature. That's why gametes are always haploid and zygotes are always diploid in nature. Now we learned the ratio of 9:3:3:1. But if you remember yesterday, I was talking about this concept that deviation from this ratio started to come up. If you remember, a few days back I explained the dihybrid test cross ratio will be always 1:1:1:1:1. So these ratios started to become deviated. That's why scientists started working a lot, and finally Morgan explained the concept of linkage, and linkage is responsible for changing this ratio as well as changing the test cross ratio. So the law of independent assortment we understood, and the law of independent assortment's exception, because I told you in the last class only Which law of Mendel is universal? Only the law of segregation. Rest all other laws have exception. So what is the exception to the law of independent assortment? Linkage. See, please remember one point. Independent assortment is only possible at two positions. That means there are two reasons when independent assortment will take place. One reason, the two genes or the two characters which a person is studying, like Mendel was studying. So when two characters controlling genes are present on non-homologous chromosome, like for example, A gene is present on first chromosome, and if you are learning about the inheritance of gene A and gene B, so by chance gene B is present on the fifth chromosome. So when two genes you are studying controlling two different characters are present on the separate chromosome or non-homologous chromosome, then 100% independent assortment will take place. Second reason, when two genes are present, because we have learned that number of genes are thousands, but the number of chromosomes are in 23, 28, 24, such numbers are there. So the number of chromosomes are very less when compared to the number of genes. That means, and we know genes are present on the chromosomes, that means there are many genes which are linearly present or arranged on a chromosome. So those two genes which are present on the chromosomes are always called as linked genes. Now the second point. First point, when will the independent assortment take place is when two genes which you are studying are present on separate chromosomes or non-homologous chromosome. Second point, when two genes will undergo independent assortment, second reason is if they are present on the same chromosome. See, there are only two possibilities. Two genes are on separate chromosome or two genes are on same chromosome. So how will the two genes present on the same chromosome separate from each other? That they are positioned, answer is they are positioned on the same chromosome at a great distance, so that every time crossing over takes place, they are going to go separate from each other. So two possibilities for independent assortment are: first, two genes you are studying are on non-homologous chromosome, or if they are on the same chromosome, then they are far enough so that every time crossing over occurs, they are going to separate from each other. Every time they are going to separate from each other.